All right, so my name is Matthew Hepworth, and I'm going to do some lesson 3 problems, practice problems with the solutions. And so for problem 1, we suppose that the PBr322 plasmid was cleaved with the restriction enzyme PST1. Also suppose that the PST1 digested segment of a mouse genome DNA was then ligated into this PSTI site and the PBr322. This recombinant DNA molecule is used to transform E. coli cells. And so for problem part B, it asks what antibiotic should be added to the medium in order for it to survive. And so I drew out the plasmid right here. And so before it was cut and the strip was added. And so as you can notice that the PSTI1 is right here in the ampicillin region. So once the enzyme is cut, once the enzyme cut and the ligation parts inserted, the ampicillin region is kind of distorted, it's separated, so you can't add ampicillin into it, or else if you added ampicillin to the plate, the plasmid, well, the bacteria containing this plasmid would die. But if the plasmid did make it into the bacteria and the plate contained tetracycline, it would grow because this tetracycline tetracycline part is still intact. So if the plasmid was incorporated into the bacteria would grow because it has tetracycline resistance. And then would the presence of bacterial colonies resistant to, and what if there's a presence of bacterial colonies resistant to both antibiotics? This means that if the plasmid, if the E. coli bacteria is resistant to both ampicillin and tetracycline, it means that this enzyme didn't cut, and then that this strain right here wasn't ligated into the plasmid, because if it was ligated into the plasmid, it wouldn't be ampicillin resistant. But since it is ampicillin resistant, we can conclude that this particular strand was not properly ligated into the plasmid, and which was taken up by the bacteria. And then if the digest happened with BAM-H1 and ECO-R1, how many fragments would they be? So if we cut right here and right here, so if we cut right here and there, then we'd have 2 fragments, because we'd have 1 fragment right here between the ECO-R1 and the BAM, and then we have another fragment right here. So we have essentially 2 fragments. Or if I did on the recombinant DNA, same thing. So we have 1 fragment right here, and then another fragment right here. And it says if we would digest with BAM and PSTI1, how many fragments would they be? And so, since we're cutting with PST1, we notice we'd have a fragment right here between both PSTIs, and then we have a fragment right here between the PSTI and this part of the BAM right here. So there's the second fragment, and then the third fragment would be between this PSTI and this fragment right here. So you'd have 1, 2, 3 fragments. So problem 2 questions concerning DNA, PCR, and DNA sequencing. So for problem 1, if you have 1 primer, what would be the problem with that? And so if you only have 1 primer, essentially here's your DNA strand right here from 5 to 3 prime and then 5 to 3 prime. If you only have 1 primer indicated right here in the purple, then only this strand would be copied. Which, if this strand does indeed contain our strand, our sense, then it's okay. But the problem is that it won't grow exponentially. You have to run it for a lot more time, like say 80 plus cycles. So instead of having the time go from instead of time doing PCR action be 3 hours, it could be upwards of 6 or 6 or 7 or maybe 8 hours. So it's just going to be a lot more time consumption, which if you had 2 primers, the time would go down because you have 2, and then that particular set that you're trying to copy with 2 primers would grow exponentially because you have 2 of them. And so it asks, next problem is how many copies of the double-stranded DNA would be made after 15 cycles. And since we're making 2, you have 2 strands after each cycle, so you take that 2, you raise it to 15, and so after that you're going to have 32,768 copies. And so what's unique about the Taq polymerase? This DNA polymerase was isolated from Thermus aquaticus. This bacteria can survive at very high temperatures. Hence, it's found in like places like Yellowstone and stuff like that, places where there are high temperatures. And so its DNA polymerase will function at high temperatures, because if its DNA polymerase didn't function at high temperatures, then there wouldn't be bacteria because they couldn't do cell replication. But since there is bacteria, we can assume that their DNA polymerase is very high temperature resistant, and so that's perfect for PCR because you need the DNA to go at a high temperature to break those hydrogen bonds, to break them apart, right? And then you need a polymerase to synthesize the DNA. So you need a polymerase that's resistant to heat, which that's the Taq polymerase it's a good option, and it really helps with PCR. And then what are the structural differences between the NTP, the DNTP, and the DDNTP? So the NTP, so I drew little ribosomal molecules right here. So for the NTP, there are just a couple structural differences. So the 2' carbon right here, which I indicated, and the 3' carbon, there are hydroxyl groups for the NTP. For the DNTP, at the 3' carbon, I take that back. I'm sorry. That's supposed to be at the 2' carbon. There's only a hydrogen group, and at the 3' carbon there should be an OH group. So I have those backed up. So this should be over here, and then that should be over there. So sorry about that, but that's a structural difference. And then the DNTPs is at the 2' carbon and the 3' carbon. There are no hydroxyl groups. So problem 3 asks about the several restriction sites. So you have a restriction enzyme with a recognition sequence, and so for each of the above enzymes, if we assume that this genotype is composed of 30% A, 30% T, 20% G, and 20% C, what is the average distance between these restriction enzyme recognition sequences? So you have this table that I drew out, that was on the slide before, along with these ratios, and so the average distance, what you have to do is, so you take each one, so A is equal to 30%, G is equal to 20. And so basically to convert to decimal form, so you have 0.3, and you have to multiply each one of them. So 0.3 times 0.2 times 0.2 times 0.3. Because 0.3 indicates to T, 0.2 indicates to C, 0.2 indicates to G, and 0.3 indicates to A. So what you do is you multiply all these together to get a certain decimal, and then you take 1 over that decimal, and that gives you the average distance it goes. So that goes about 278 base pairs according to this problem. So there's the average distance between each of these restriction enzyme recognition sequences. So this one's 278 base pairs. So following the same format, so GGCC, it was all about 20%. So you take 0.2 times 0.2 times 0.2 times 0.2 to get 0.0016. And now you take 1 over that and you get 625. And then for part C, you follow the same format to get 3.24 times 10 to the negative 4th. And so 1 over that is going to be 3,086 base pairs. And then for part D, continue to follow the same format. 0.2 times 0.2 times 0.3 times 0.3 times 0.2 times 0.2 is equal to 1.44 times 10 to the negative 4th, which 1 over that is 6,944 base pairs. And then the knot, follow the same format to get 2.56 times 10 to the negative 6. Take 1 over that, and you have 390,625 base pairs. For problem 4, we use DNA sequencing to determine the nucleotide sequence of the molecule shown below. So you have 5' to 3', you have CC, GAA, TAC, TAG, TTC, and these are the primary nucleotide sequence. So after DNA sequencing using the Sanger method described in class, it was separated using electrophoresis. What should the banding pattern look like? And so you have the complementary sequence, which goes with the primer. So you have GG, or 3' to 5', you have GGC, TTA, TG, AC, AAG. And so using the Sanger sequence, you want it to look like the complementary sequence. You have the DDGTP, the DDATP, DDCTP, or DDTTC, and the DDCTP. And so you just kind of following this complementary sequence right here. So you have G, G, and then you have C, and then you have T, T, and then you have A, and then you have T, or T, then G, and then A, C, and then A, G. And it's all going from the negative electrode to the positive electrode because these molecules are more negatively charged. And so as you have the positive electrode, they're going to travel to the gel in that fashion. So question 5 refers to the restriction mapping. So it talks about suppose that circular plasma was cleaved with 2 restriction enzymes, BAM and ECO. The DNA fragments obtained were the following. And so these are the so you just cut with BAM, these would be the sequences. If you cut with just ECO, these would be the sequences. And if you cut them with both, these would be the fragments. And so draw an image that represents it, and then part. And so sorry for that pause. So we have to draw the image of as it represents the gel electrophoresis, which is shown right here on the left, the restriction mapping. So if you have this the BAM, I drew a little key right here, so 4,000, 3,000, 2,000, 1,000, 500, 100. So the BAM is going to cut at 24, 12, or 2,400, 1,200, and 400. So I try to draw representative bands as shown right here for the BAM. And the ECO is just going to cut at 4,000, so it's going to show up right there. And the ECO with the BAM, they're going to be bands at 1,600, 1,200, 800, and 400. So here's a band at 1,600, 1,200, 800, and 400. So these are how the banding patterns would look. Then for part B, it says draw a linear fragment. 7,500 base pairs is cleaved with the individual restriction enzymes, and then and so you have the HIND 3 is cut at 25. You have a base fragments of 2,500 and 5,000 base pairs, and then you have the small, which is you have fragments of 2,000 and 5,500, and then you have the HIND and the small, which cut together. You have fragments of 500, 2,000, and 5,000. So as it's shown right here, that the HIND plus the small, you're gonna have fragments of 500, 2,000, and 5,000. And so since the small cuts at 2,000, you're gonna have that one first, right? And then there's gonna be a small fragment of 500, and then from like the very beginning, the HIND is gonna cut at the 2,500 base pair mark, so it's gonna cut right here. So that's how you have 2,000, 500, and 5,000. And then for part C, it says this, we're gonna draw a circular plasmid map. And so the claw, it cuts at 5,200 base pairs, or it's as a fragment of 5,200. The sac is going to have fragments of 2,600, 1,200, 800, and 600. And the claw with the sac is going to have fragments of 1,400, 1,200, 800, and 600. And so we need to draw a circular map. And so I draw a circular map. And so what I did is I started down here with the sac. So I did sac. I'm like, okay, so sac to sac, it's going to have a 600 base pair fragment. And then I did another sac to sac. I'm like, okay, that's another, because sac cuts at, it doesn't have base pair fragments of 600 and 800. And so that's how I got those two. And then I did the same thing. So I did another sac to sac, and I got another 1,200 base pair right here. So adding these up right here, you have 1,200 plus 800 plus 600. That comes out to about 2,600 right there. So I brought halfway. And then what I noticed is that from this sac to this sac, it should produce if the claw wasn't present, it would produce a fragment of 2,600, which is indicative right here. However, with the claw and the sac together, it could produce a particular fragment of 1,400 base pairs. And so that's why I do a sac to a claw, which indicates 1,400 base pairs. And then so I added these up. So I added this 26,000 to this 2,600 to this 1,400, and I got 4,000. And I'm like, okay then, so that's 4,000. Then this remaining fragment has to be 1,200 because it has to equal 52 base pairs. And these are the problems and the solutions of lesson 13. Thanks for listening.